Our recent publication actually got a significant exposure. It was a very elegant study that was published in Science. In this study, we took cancer patients in which there is evidence that they have killer cells of the immune system penetrating into their tumors. This is a strategy that is known with a professional name of adoptive cell transfer. Why adoptive? Because then the surgeon is taking the tumor out from the patient. In the lab, people extract these killer cells from the tumor and now expand them to huge numbers. And afterwards, give these killer cells back to the patient. Okay, this is a strategy that was developed in Europe, in Belgium, and in the US at the National Cancer Institute. And in Israel, there is only center outside the US that is developing this strategy is in Israel, in Tel HaShomer Hospital. Now, the major problem with this strategy is that when you take a tumor and you extract from this tumor the killer cells, only 20% of them respond to the tumor, and 80% don't. And we ask why 80% of these killer cells don't respond and 20% respond. Now, we discovered that actually these killer cells are not a single population of cells. There are hundreds of subpopulations of cells that exist there in the tumor, and then it's very difficult to analyze them because a human being can look only at 1, 2, 3, 4 parameters. But now we've collected more than 90 different parameters, 90 different surface markers that exist on these cells, which actually the ID card of these cells. And the ability to analyze and actually try to understand what is the difference between the cells that are responding and the cells that are not responding, these differences can be looked only when you apply computational tools. And this is why we call this area system biology, because we take many parameters, many complicated questions, and we use tools related to physics and computer sciences. So this is a great idea of a PhD student in my lab and a PhD student in the Department of Computer Sciences. And they actually used an algorithm that they've been developing to analyze and see whether the responding cell has a particular signature which makes them unique compared to the non-responding cells that have a different signatures that makes them unresponsive or not reactive. And eventually they could find a signature that is composed of between 5 to 7 molecules that are sitting on the surface of the cells and can actually predict by an accuracy of more than 95% whether a particular culture that we take from the patient will be responsive or not to the tumor. So this was great results because we actually, each patient that is being considered for treatment, we can take a sample and predict using this algorithm whether it is whether it is capable to respond or not to the tumor. And then we took the project even one step further. We asked, can we, by identifying this signature, can we transform a non-reactive culture of killer cells into a reactive one? And we were able, in 10 cases out of 12 that we've tested, so we took 12 patients, and in 10 out of 12 patients, we could take killer cells from their tumors, which were not reactive to start with, and transform them by manipulation of the composition of subpopulation in this mixed population. By manipulation, we could transform them from a non-reactive into a reactive population. So this generated a very appealing results, which are now we are continuing, and actually we are hoping to start with appropriate funding because this was submitted to a large European consortium of people doing immunotherapeutic studies. We hope also to bring here something to the clinic, being able to translate it into more efficient studies in the clinic, because now we can not only predict which culture of killer cells will be more reactive against the tumor, but also to manipulate these cells and hopefully to cause them to be more potent against the tumor. So this is our latest hot stuff that we are working in the lab and looks very promising.